thank you also for the committee for giving me the opportunity to present my work today, and also HFSP for financing my postdoc that I'm doing in Didier Stenier lab in Bad Nauheim. For those who don't know, is close to Frankfurt, and it's at the Max Planck Institute for Heart and Lung Research. And I'm going to talk about live imaging of translation during zebrafish embryogenesis. So one of the most fascinating question in developmental biology, at least to me, is how a single cell can give rise to such complex organism. And from invertebrate here, and Drosophila to vertebrate, here an example with zebrafish, embryogenesis is very dynamic and highly coordinated in time. And during this process, the cells will have to divide, to move, and progressively acquire specific identities. And this organization depends notably on gene expression programs that are tightly regulated in space and time, as illustrated here with in situ hybridization in purple of many different mRNAs. And so it shows the variety of pattern of expression corresponding to different cellular identity. And while I guess most of you know, the central dogma of molecular biology defined at least 60 years ago, most of our knowledge about how gene expression is regulated in space and time rely on the dynamics and coordination of transcription in developing embryos. But the dynamics and the coordination of translation is way less understood, and this is the step I'm interested in. And so the big question I have is to understand how regulation of translation can contribute to the global organization of the embryo as an independent layer of regulation. And so when I first thought about organization of the embryo, one key family gene that came to my mind are the morphogen that are involved in the formation of embryonic axis. And so I got interested in one of them, which is the bone morphogenetic protein 2B. So this protein is a secreted ligand involved in epidermis and mesoderm specification, as well as bone and muscle cell differentiation. And so this gene is expressed as a gradient in the zebrafish embryo, as you can see here, from the ventral side to the left to the dorsal side on the right. And so as you can see, it starts on the ventral side, and then at 6 hours post-fertilization, we start to have an expression at the most dorsal part. Sorry, I'm not trying the pointer because I think it's too complete. And so what is really interesting also with this protein is that the quantity is critical for development. Indeed, if you mutate the gene, you have dorsalized embryo, and if you overexpress BMP2B, you have a ventralized embryo. And when we looked at what has been published in bulk analysis, looking at translation efficiency, so they basically compare ribose-seq with RNA-seq, and what we saw is that BMP2B is one of the with the lowest translation efficiency. And so this led us to ask how such an important gene is regulated at the level of translation, and what determined this low translation efficiency. So the question then we had is how BMP2B translation is regulated in space and time. So what is this, its fine regulation? And so for this, we used a live imaging approach to be able to live image translation in zebrafish embryo. So the tools has never been made before, and so we used first the gold standard method, which is the SunTag system. So this system is based on a SunTag here in purple that you insert repeats into your gene of interest. And upon translation, these SunTags are going to be recognized specifically by a monochain antibody fused to a GFP. And so so this you can encode it into the genome, and then it will create accumulation of GFP at the site of translation at the mRNA, so at the single molecule. And so this will create bright foci above the background. So actually, this SunTag system didn't work in our hand, in zebrafish. So that's why we turn to an orthogonal tagging system, which is called the alpha array. So it's the exact same principle as the sun tag, but this time is called alpha tag. So we multimerize, put repeats in the gene of interest, and then it will be recognized by a nanobody fused to a GFP. So in our case, we tag the BMP2B with the alpha tag. And so then we had to create two different zebrafish strains, one expressing the nanobody fused to the GFP in an ubiquitous manner, and one expressing the BMP2B fused with the alpha array. So then I crossed these two fish. So using this, it's gonna work? Yes. So using these mating tanks, as we call sex on the beach, because the zebrafish like it on the beach. And so we then injected the embryos that you can hopefully see going to the bottom. So we then took these embryos, inject them with, depending on the different construct we wanted to use. Then we waited for an hour for the embryo to recover. And then we used the lattice light sheet imaging. So after many trials on different microscopes, we finally found one that was suited for this type of imaging. And so we could image for the first time nascent translation in vertebrate embryo, so here in zebrafish. So the dots that you see bright here in the cytoplasm, so the small ones that are moving rather slow. So they represent mRNA, single molecule of mRNA in translation. And so thanks to also this lattice light sheet imaging, we could have a wide field imaging for the embryo of zebrafish, and also a high temporal resolution because we have 1 second per z-stack for the whole embryo. And so then, this single mRNA in translation, we can track them during time. Yeah, it's not moving, it's okay. And so then we can measure the fluctuation of the intensity of a single mRNA in translation. And this fluctuation of intensity is the reflect of how many ribosomes you have on your mRNA. So how many are coming, how many are leaving. And so together with a physicist in Timothy Stasevich's lab at the Colorado State University, Tatsuya Morizaki, we could autocorrelate this signal and then find translation kinetics such as the initiation rate of ribosome or elongation rate. And so we could then compare different constructs. So in this case, we compared the stable line expressing the BMP and the transient injection of BMP tagged. We could find the same initiation rate. But then when we compare with a different 5' UTR, so we switch the 5' UTR of BMP2B with an exogenous 5' UTR, the one of the virus Zika. This time we could increase the initiation rate of ribosome at least 3 times, meaning that BMP2B translation appears to be below its maximal initiation capacity in vivo. So it means that BMP2B is regulated at the initiation level. On the second hand, we of course wanted to know if all the mRNAs were in translation or not. So for this, we combined mRNA and translation imaging in the zebrafish embryo. So we used the alpha array, and then we used another technique to visualize this time the mRNA. So we put it MS2 loops. So these loops, when they are transcribed, they will be recognized specifically by MCP, fused to the GFP this time. And so we could then do the dual color imaging. So here in purple, you have the mRNA, and in the translation is in blue. And so we could see both mRNA and translation at the same time moving in the embryo, and we could measure the percentage of mRNA in translation, which appeared to be rather low for BMP, so around like 25% of mRNAs were in translation, meaning that only a subset of BMP2B mRNA are actively engaged in translation in the embryo. So what I've shown you so far, so we saw that BMP2B is regulated at the level of initiation of ribosomes, but also at the capacity of its mRNA to be in translation, so the percentage of mRNA. And so we also tested another feature of the mRNA that could influence translation. And so I'm going to just show you one example here, and I'm going to say cut the long story short, but basically what we did is to test the capping of the mRNAs. So the mRNAs, they are normally capped with a guanine, and then we replace this cap with a cap analog that is non-functional, so it cannot be recognized by all the translation factors. And so we in vitro transcribed these two mRNAs, we injected them in embryos expressing the nanobody alpha, and then, to our surprise, we saw that BMP2B, even without a functional cap, can still translate. And actually, it can translate at the same efficiency, so we saw less dots, but still the single mRNA molecules, they were able to translate at the same rate. And this seems to be UTR specific, because if we change the UTR with a housekeeping gene UTR, so the one of actin, of BMP2B, this time we abolish the translation of our reporter. So meaning that there is non-canonical translation that seems to be specific to some 5' UTRs, in this case BMP2B. So what I show is that BMP can be translated in a non-canonical manner, so cap independent. And we actually hypothesize now that this mechanism contributes to the robustness of development. So like when there are like some phenomenon during development where canonical translation is reduced, maybe this mechanism of non-canonical mechanism can take over for certain gene and still allow translation of such important gene as BMP2B. And so this event can be during viral infection, where it's known that canonical translation can be reduced, also stress or apoptosis. And so we actually saw that there is a short time window during the early development of zebrafish where mTOR activity is reduced, so then canonical translation factor seems to be reduced. So maybe this non-canonical translation of BMP can be sustained also during these early steps of development. So the conclusions are relying in 3 bullet points. So we first did for the first time live imaging of translation in developing vertebrate. We are now trying to look at later stages if this is possible. We also show a fine analysis of translation that is now possible. And we are actually now trying to do this analysis in spatial resolution, so within the BMP2B gradient. And then this non-canonical mechanism for translation in the embryo, we are now trying to identify the factors involved. And so with this, I would like to thank Didier Stenier, my postdoc supervisor, also all the people who got involved, in this work. Also, of course, HFSP, the collaborators, Timothy Stasevich and Jérémy Dufour. And so, yeah, so right now, I'm moving a postdoc position in Bad Nauheim to a permanent position that I just got at the CNRS in Montpellier to join the lab of Edouard Bertrand. And so this is, I also would like to thank HFSP for supporting me for this transition. Thank you for your attention.